I ended the last video saying I wanted to discuss cortisone and cortisol relationship in COVID-19. This topic gets into the question, how does beta-hydroxybutyrate affect the immune system? It also leads to the question of natural ketosis versus ketosis in a bottle. Do you have to be in ketosis with all the natural processes of ketosis for exogenous ketones to work? Or can you ignore diet and just take ketone ester or MCT oil or ketone salts? There's a third question we ask in this video. Do steroids improve survival in treating COVID-19? The first two questions are asked and answered in a paper out of Yale University. The labs of Vishwadeep Dixit and Akiko Iwasaki collaborated on a study of ketogenic diet in mice exposed to a dose of influenza A virus. The principal author of the paper was Emily Goldberg, who is now at UCSF. This is a seminal paper in the use of ketogenic diet for infectious disease, and you won't find a finer written paper. Dr. Goldberg's team used a mouse model of the flu. The flu is caused by orthomyxovirus. The mouse was an inbred line C57 black 6. As an inbred line, C57 black 6 has lost two important genes. One gene that provides resistance to the flu, the myxovirus-resistant protein 1, or MX1. The other is NNT, nicotinamide nucleotide transhydrogenase, which we will revisit later. The C57 black 6 mouse had its MX1 protein phenotype restored by breeding in Dr. Iwasaki's lab. Mice on a ketogenic diet for 7 days were protected from influenza A virus. They did not lose weight as fast as chow-fed mice. More of the ketogenic mice survived. The ketogenic diet mice had lower titers of virus in their lungs. Why did this protection occur? Dr. Goldberg took lung cells from the keto diet mice and the chow-fed mice and used transcriptomics to identify differences in mRNA between the ketogenic diet and the chow mice. The data was fed to a search tool for ingenuity pathway analysis. They expected to find signaling pathways such as interferon or interferon-stimulating genes. They saw no signs that interferon was working. They did, however, find that there were a lot of T-cell regulation pathways. A volcano plot revealed that the gene for T-delta gamma receptor was among the top 10 overexpressed genes in the ketogenic diet-treated lung tissue. They looked in a second database, the Immunological Genome Project, and found other genes that correlated with gamma-delta T cells, and these were found being expressed in the lung cells of the mice fed a ketogenic diet, included CXR6, BLK, CD163 pipe 1, and CCR4. So they found that ketogenic diet protected mice from influenza A virus and that gamma-delta T cells were involved. Then they asked if it was due to fat or ketogenic diet. And finally, they gave the mice an exogenous molecule that turns into 50% active R3-hydroxybutyrate or D-beta-hydroxybutyrate and 50% inactive or S or L forms. I say inactive, I am only referring to the selectivity of beta-hydroxybutyrate dehydrogenase, which converts NAD plus into NADH to feed the electron transport chain and is specific to the R or D form. The answer Dr. Goldberg's team found was that racemic 1,3-butanediol did not have the same result as a ketogenic diet. According to this experiment, one could not use ketosis from a bottle under their condition. The group from Dr. Dixit's and Dr. Osaki's labs posted supplemental data that showed perhaps there was something going on with glucocorticoid receptor pathway. Dr. Seed suggested we look for alternative explanations. One of the alternative explanations involved NADPH, NAD8, and the way they drive cortisol-cortisone interconversion. The glucocorticoid receptor responds to cortisol. Unbound cortisol can be converted to the inactive form, cortisone, by type 2 11-beta-hydroxysteroid dehydrogenase. Type 1 11-beta-hydroxysteroid dehydrogenase converts cortisone to cortisol. One of the ways they work is based on specificity of NADPH for type 1 and NADH for type 2. So when you have high levels of NADPH, cortisone, inactive, is converted to cortisol, which is active. When you have high levels of NAD+, cortisol, which is active, is converted to cortisone, which is inactive. Cortisol downregulates the immune response. It is thought that too much cortisol early in the infection would suppress the acquired immune system before it had time to work. If you wait until C-reactive protein and interleukin-6 start going way up, you can give cortisone analog drugs and they will blunt the immune response, helping to turn off the cytokine storm. So the question arises, should you give steroids to people with COVID-19? Several studies and metadata studies of studies indicate that if you are on oxygen or other respiratory support, steroids will increase your probability of survival. However, if you are not on oxygen, steroids may increase mortality. So we've seen two ways that ketosis can alter the immune system: by raising gamma delta T cells and by affecting the cortisone-cortisol balance.